Okay, so we're going to start now. Okay, talking about these parts are more to very factual. Okay, we have to explain what are the learning outcomes that we need to learn here. We need to explain the advantage of producing human proteins. Okay, produce a human protein using the recombinant DNA that we have learned. Okay, and particularly we talk about the insulin factor 8 for the treatment of hemophilia, and adenosine deaminase for treatments of to use it for the treatments of severe combined immunodeficiency, or we call it a SCID. So those disease I will go through with you guys. Like hemophilia I won't, but SCIDs, yes, I will go through with you guys. Okay, so this will be the first part. So first part, basically we talk about the recombinant DNA used in the medical as a medicines here. Okay, and then we need to outline the advantages of do the screening for the genetic conditions. For example, test for specific genes such as breast cancer, BRCA1 and BRCA2, quite famous, these two genes. Okay, because of the one of the celebrities, Angelina Jolie, okay, went for this mastectomy, remove the breast because she inherits these mutated copies of the BRCA1 or BRCA2, I can't remember, but basically that is one of the way. Okay, and also gene for hemophilia, sickle cell anemia, Huntington disease, and the cystic fibrosis. Okay, so the disease Huntington, I don't need to go through sickle cell anemia, hemophilia, and cystic fibrosis. Those are the diseases that we have. Look at it in our previous topics. It had to change. Okay, so please refer back. And outline how genetic disease can be treated with the gene therapy, and discuss the challenges in the choosing appropriate vector, because we cannot put the gene straight away. We need to use the viruses, liposome, naked DNA technology, reference to the SCID, and also the cystic fibrosis. I'll focus in these two, okay, SCID and also the cystic fibrosis for our syllabus. Okay, how we use the gene therapy. And last, we look at the social and ethical considerations of those gene testing and the gene therapy in medicines. Okay, so we're going to look at these IVF, embryo biopsy, and pre-selections, and the therapeutic abortion. Those are the idea that we're going to look at it. So basically, the second part of the topics, this part will be the genetic screening for the genetic disorder, and the last part of these subtopics will be gene therapy. Okay, so I grouped three of them together, okay, and put it under genetic technology applied to the medical fields. Okay, so we're going to start with recombinant proteins, okay, for medical programs, medical field. So genetic technology allowed the recombinant protein specific to human to be made. This protein included insulin factor 8, adenosine deaminase, human growth hormone, thyroid stimulant hormone, so many, but we only focus in these three. Okay, insulin factor 8 and adenosine deaminase. So highlight this. This is a very famous question always asked. Okay, what are the advantages of genetic engineered humans insulin? Okay, so why we use the genetic human insulin rather than the animal insulin? First, indistinguishable, is identical. Basically, it's identical. We can use the word because from human, okay, from the natural human insulin produced in the pancreas, and therefore less chance for immune response to target and remove the genetic engineered human insulin from the blood plasma, so it can stay longer. You try to imagine if you use the animal insulin, this animal insulin will act as the non-self antigen. So if it act as non-self antigen, what will happen? Your immune system will remove it. So means that if I inject 100 milligram, at the end maybe only left 5 or 10 milligram there. Why? Because your immune system will remove this animal protein because it acts as a non-self antigen. And because it's a genetic engineered human insulin, means identical, so it's less likely to cause the allergic reactions in the diabetic patients. Okay, because identical. Human insulin receptor have more rapid response because the binding effect of affinity will be better. So basically means that the binding effect better will give us the rapid response. Okay, and the mass production of genetic engineered human insulin is possible because as we know that E. coli actually require very simple nutritional requirements. Okay, and also if we use this genetic engineered human insulin, basically means that we can reduce the risk of transmitting animal diseases. Cry not, okay? And also fewer ethical and moral and religious issues when genetic engineered insulin is used. Why? Because, as I say that, if we use this bacterial, we don't kill the animal, right? We don't kill the animal. Okay, in terms moral, ethical is more accepted, and religious issue as well. Some may say that, okay, I don't want to use the animal product. Okay, so this is the first part. So please highlight this activity 16.24. This is the common question asked in the essay question, and also for the structure. Okay, what are the advantages? Why we use the genetic human insulin rather than other types of the insulin, for example, from the pig. Okay. Now look at this recombinant factor 8, okay, for the blood clottings, okay, hemophilia. So we do know that hemophilia is inherited disorder, unable to synthesize factor 8. So where's the factor 8? If you look at this, factor 8 here, activated factor 8, we're going to activate factor, okay, 10, and eventually we can get the fibrinogens, okay, to be converted to fibrin and form the clot. Okay, so for us, we do know that hemophilia is you can't have the proper blood clots due to the clotting factor 8. So what we're going to do here is we can use the recombinant factor 8, okay, but slightly different for recombinant factor 8. After trial for so many rounds and try and error, scientists found that hamster kidney and ovarian cells will be the best to culture, okay, to produce the factor 8. Are you clear? So no question will ask why hamster kidney or ovarian cells. Basically, they are eukaryotic cells, so they give us the most optimum products. Okay, like insulin, we use E. coli, but doesn't mean that for other type protein must be E. coli. So in this case, we use hamster kidney and also the ovarian cells, so it gives us a better product. So what is the benefits here? Now, previously without this, without the availability of the recombinant factor 8 is isolated from donated blood. As at that time, we do not clear about HIVs, okay? So there is a risk of the infectious, okay? But recombinant factor 8 actually avoids this problem, okay? Then the third recombinant proteins that we use here is the recombinant adenosine deaminase, or ADA. Okay, so before we go to the ADA, we're going to look at this, what we call a severe combined immunodeficiency. It's a type of immunodeficiency. The symptom will be similar, quite similar to this HIV infection, also immunodeficiency, but slightly different here is this is a severe combined. It's a group, it's a class of the disease. Okay, it's characterized by the disturbed development of the functional T cells and B cells caused by the genetic mutation, not because of the HIV. It's a severe combined immunodeficiency. So in this case, because they are immunocompromised, so therefore they are very vulnerable for, okay, or susceptible for the bacterial, viral, or even fungal infections. Okay, so in this case, the skin is also known as the bubble boy disease, because in these kids have to be isolated in this bubble boy. Can you see that? Because the supply of the air must be a clean air, a sterile condition, so that he won't fall sick or get the infections from the bacterial, virals, and also the fungus. Okay, so there are many forms of the SCIDs, okay, severe combined immunodeficiency. One of it, the form is because of the defective enzyme called adenosine deaminase. Adenosine deaminase. So this adenosine deaminase is the one that necessary for the breakdowns of the purines. And because of the ADA, okay, lack of this ADA enzyme, it causes accumulations of the nucleotides. Can you see that? We cannot break down of DATP. So this metabolite will inhibit the activity of the ribonucleotides reductase, the enzyme that reduces this ribonucleotide to generate deoxyribonucleotide. Okay, so means that if we cannot generate deoxyribonucleotide, means that what will happen? The effectiveness of immune system depend on the lymphocyte proliferation. Okay, what will happen here is now, in order to get your T cell to make many T cells, okay, they need to carry out mitosis. But before mitosis, they need to carry out DNA replication. Because of accumulations, okay, we cannot, okay, accumulations of this DATP, we lack of this. Are you clear? We lack of this DNTP. Sorry, DNTP. Are you clear? Okay, in this case, deoxy. So if let's say we cannot generate this deoxyribonucleotides, what will happen? DNA replication is inhibited. Okay, so what you need to know, these are the extra informations for you guys, for informations only. Okay, so what you need to know basically here, lack of this adenosine deaminase, it will cause the lymphocyte proliferation to be inhibited. So means that your T lymphocyte cannot form the clones. Therefore, immune system is compromised. Very simple for you guys. So what is the effect? Without this, or with this defective enzyme, the adenosine deaminase, therefore T cells cannot proliferate. So therefore, the particular individual will have this immune system to be compromised. Okay. So how? In this case, the lack of enzyme, we give them the enzyme. So this enzyme can be made, okay? So can be synthesized using the recombinant DNA technology. So this recombinant DNA technology, we can make it genetically modified insect larvae. Okay, so for example, we can use a cabbage looper moth capital. Okay, so use insect larvae. So the question here is, okay, why? Okay, you don't need to answer very specific. Basically, they are eukaryotic cells. They give us, okay, they can help us to fold the ADA into precise three-dimensional shape. Okay, better compared with the E. coli. Okay, so enzyme is used for the patients that are waiting for genetic therapy, gene therapy, or for the patient that gene therapy is not possible. So gene therapy, we're going to discuss it tomorrow. Okay, for gene therapy, we're going to discuss it tomorrow. So,